Hello, I'm Alicia Kowaltowski. I'm a specialist in mitochondrial bioenergetics, and today I want to talk to you about when and how to measure mitochondrial inner membrane potentials. So if you look in the literature, such as a PubMed search, you can see that more than 3,000 papers are being published a year in which there are measurements of mitochondrial inner membrane potentials. And I think this reflects a general interest of the public in mitochondrial phenomena, which have proven over the years to participate in many different biological phenomena. And people are really interested in measuring what's happening to mitochondria, and therefore lots of measurements of mitochondrial inner membrane potentials happen. Now, when I look at the literature, many of the papers that are assessing mitochondrial inner membrane potentials are actually trying to measure something that they call mitochondrial function. Now, I'm saying mitochondrial function within quotes because mitochondrial function, for a person who studies mitochondrial biology, is just about everything. There are many different mitochondrial functions, which include oxidative phosphorylation and ATP synthesis, include calcium regulation, include the production of oxidants, production of different steroids and different lipids from membranes, many different mitochondrial functions. And in that sense, I really want to stimulate all of you to read this wonderful paper that was published by Anna Monzel, Tonio Enriquez, and Martin Picard, in which they review the fact that mitochondria have different features, have different activities, including the mitochondrial inner membrane potential, many different functions, many different behaviors. And if we group all this into a blanket term of mitochondrial function or mitochondrial dysfunction, what actually may happen is similar to the description of the blind man describing an elephant. What may happen is that we're all measuring different things, seeing different aspects of mitochondria, but putting them under the same blanket term of mitochondrial function. And therefore, we're really losing specificity that's important to understand all of these biological phenomena that are controlled by different characteristics of mitochondria. So I want to really stimulate people to be a lot more specific than talking about mitochondrial function, especially when they're measuring mitochondrial inner membrane potentials that really aren't a mitochondrial function per se. Now, when I read papers that measure mitochondrial inner membrane potentials, most of them are measuring these inner membrane potentials as a proxy for oxidative phosphorylation, which is perhaps the more canonical of the mitochondrial functions. And here, if you take anything from this video class, I would like you to take that measuring mitochondrial inner membrane potentials is not a good way of assessing mitochondrial oxidative phosphorylation. First of all, for a purely theoretical reason, which is the sensitivity of the changes in membrane potential versus changes in oxygen consumption rates, for example, as a measurement of oxidative phosphorylation. So what we have here is a cartoon of the expected changes in oxygen consumption rates without and then with oxidative phosphorylation by adding ADP. So when you're measuring oxygen consumption rates in mitochondria and you add ADP, this is an isolated mitochondria, this is done quite frequently when you're measuring mitochondrial respiratory control ratios. What you're going to see is an increase in oxygen consumption rates, which can vary by at least 2 times the oxygen consumption rate you had originally, to maybe even 15 times faster oxygen consumption rates in the presence of oxidative phosphorylation versus in the absence of ADP, therefore the absence of oxidative phosphorylation. Now, on the other hand, if you're measuring mitochondrial inner membrane potentials and you add ADP stimulating oxidative phosphorylation, you're going to have a decrease in membrane potential, which may be around 20% of the original membrane potential. It's not going to be more than that. Therefore, from a theoretical standpoint, if you're going to see changes in oxidative phosphorylation, membrane potentials are going to be a lot less sensitive than oxygen consumption rates. And that's because of the characteristic of the nature of what you're measuring. Membrane potentials are basically maintained in steady states in mitochondria, while oxygen consumption is a measure of flux, and therefore it's a lot more sensitive. So just as a quick refresher, the mitochondrial electron transport chain is going to transport electrons to oxygen, generating water from this oxygen, and that's what generates oxygen consumption in mitochondria. This electron transport is coupled to proton pumping into the space of the crista. So this is typically shown in textbooks as proton pumping into the intermembrane space. But we know for the last few years that the membrane potential in mitochondria is mostly within the crista because of the location of the electron transport chain and the location of the ATP synthase. So this proton pumping across the inner membrane is going to generate a membrane potential within these crista. And this proton now has a tendency to return through the ATP synthase coupled to ATP synthesis. Now, because this return of the proton, either through the membrane or through the ATP synthase, has resistance, this generates a membrane potential. This generates voltage across the inner membrane. And in this sense, mitochondria are very, very similar to electrical circuits, in which you're going to have electrons circulating around and you're going to have a resistance generating voltage in these electrical circuits. And the voltage is going to be regulated by electron transport and also regulatory of electron transport. If you have a decrease in voltage across the inner membrane, this facilitates all these electron transport reactions and facilitates proton pumping into the intercrystal space. And this makes the membrane potential decrease a lot less than the increase in the transport of electrons. So the measurement of current, the measurement of flux, which is the measurement of oxygen consumption, is always going to be more sensitive than the measurement of voltage when you are assessing oxidative phosphorylation. So once again, I really want everybody to remember that membrane potential measurements are not sensitive and they're also not comprehensive assessments of oxidative phosphorylation. If you want to measure oxidative phosphorylation in your cells and in mitochondria, a much better measurement is a measurement of flux, such as oxygen consumption flux. Now, having said that, there are situations in which measuring membrane potentials make sense. First of all, situations in which your biological question actually requires you to measure mitochondrial intermembrane potentials. Another situation in which measuring membrane potentials makes sense is a situation in which you want to look at subcellular markers of mitochondrial oxidative phosphorylation. We really don't have good commercial and easily available mechanisms to measure oxidative phosphorylation in different parts of the same cell, for example. And under those conditions, it could make sense to measure intermembrane potentials and look at this across a cell. Now, most intermitochondrial membrane potential measurements that I see in the literature are measurements that are done in situ, mostly in intact cells, but sometimes in tissue slices. And therefore, I'm going to talk about in situ mitochondrial intermembrane potentials and what you have to be careful when you're doing these measurements, if you actually have to do in situ mitochondrial membrane potentials. Most of these membrane potential measurements are in intact cells, although there are a few in situ mitochondrial intermembrane potentials measurements that are done in permeabilized cells. Permeabilized cells are cells in which you have removed the integrity of the plasma membrane, and therefore you are assessing mitochondrial intermembrane potentials in the mitochondria in these permeabilized cells. This makes things a lot simpler because you're removing many of the artifacts and many of the complicating biological effects of having a membrane potential across the plasma membrane. And this also allows you to use non-permeable probes to the plasma membrane, such as safranin O, which can be readily calibrated. And this is a lot simpler to do than intact cells. But most measurements are done in intact cells. And here I really have to call your attention that these systems are very complex, and therefore not all phenomena measured will be generated by mitochondria. You have the whole cell there. And this is really important because there are a number of probes and a number of kits being sold in the literature as mitochondrial intermembrane potential kits or mitochondrial intermembrane probes. And they rarely tell you of all these complicating factors in which there are all these other measurements that you're doing within the cell that you really have to be careful with. So typical measurements that you're going to see in the literature are going to involve these probes. And these probes will accumulate under the specific conditions that you should be working in mitochondria in a Nernstian manner, dependent on the membrane potential. So the higher the membrane potential, the more these probes that have positive charges and can transverse membranes should accumulate within the mitochondrial matrix, stimulated by these inner mitochondrial membrane potentials. So what kind of probes do we see in the literature? Some of the probes that are used very often are precipitating probes. Examples are JC1 and JC10. These probes are going to accumulate within the mitochondrial matrix, and they're going to have a change in their fluorescence spectrum, so a spectral shift when they aggregate and even precipitate within the mitochondrial matrix upon this accumulation. I really am very critical of the use of this class of probes. The problem with these probes is because they have one color of fluorescence when they're outside mitochondria, not aggregated, and one color of fluorescence when they're inside mitochondria and aggregated, is that you really can't calibrate these probes. You can't do a relative quantification of the probe inside mitochondria and outside mitochondria because there's a spectral shift, and therefore they have different colors inside and outside of mitochondria. If you can't quantify them inside versus outside mitochondria, you cannot calibrate inner membrane potentials, because inner membrane potentials can be calibrated through the Nernst equation by comparing the concentration inside mitochondria and outside mitochondria. And if you can't calibrate mitochondrial inner membrane potentials, you are very amenable to having all sorts of artifacts in your membrane potential measurements. And we're going to talk about some potential sources of artifacts after. So I really want to stimulate everybody not to use these precipitating probes. I think the only thing they're really useful for is to make pretty pictures because they have two different colors when you're staining cells, but they really aren't good for quantifying mitochondrial inner membrane potentials. Another class of probes are probes that are going to accumulate within mitochondria, but they're not going to have this fluorescence spectrum shift. They're going to accumulate in mitochondria, and then they're going to have a change of fluorescence of the whole cell because of this accumulation only. Most of these probes are rhodamine derivatives, and some of the most used ones are TMRM, TMRE, and rhodamine 1,2,3. Basically, these are molecules that are quite lipophilic, so they can transverse membranes, but at the same time, they have a positive charge, so they're going to be attracted to that matrix of mitochondria when the mitochondrial membrane potential is high because of the negative charge of the matrix. Now, one thing that you always have to remember with these probes is that these probes in intact cells are going to be accumulated inside the mitochondrial matrix in a manner dependent on the mitochondrial inner membrane potential, which is in the ballpark of 150 millivolts, maybe 200 millivolts. But these probes are also going to accumulate within the cytosol in a manner that's dependent on the membrane potential of the plasma membrane, which can be of 60 to 80 millivolts also. So the concentration of the probe in the cytosol is going to depend on the membrane potential of the plasma membrane, and the concentration of the probe in the matrix is going to depend on the membrane potential of mitochondria and how much probe is in the cytosol, which of course depends on the membrane potential of the plasma membrane and how much probe you put outside. So you always have to think of these two membrane potentials. And very often people in the literature will forget that the plasma membrane potential will affect measurements and will affect how much probe is inside mitochondria. There are other things that can affect how much probe is inside mitochondria. First of all, time. Different probes will equilibrate across membranes at different time points. Also, there are multi-drug resistance transporters that can throw probes out of the cell, and this may change the amount of probe within the cell. Different cell types have different amounts of this activity, so you have to keep this in mind. Another thing that I really want to caution people that is often not checked in the literature is that these probes are going to accumulate within mitochondria many times more than they're added outside the cell. So even if you have very low concentrations outside the cell in the nanomolar range, the amount of probe in mitochondria can be very high. And this excess probe in mitochondria can affect the functions of mitochondria, the many different functions of mitochondria, and very typically may affect oxidative phosphorylation. So many of these probes are known in higher concentrations to inhibit specific respiratory complexes, or even to affect the membrane potential itself, affecting oxidative phosphorylation. So always try to use the least amount of probe that works for your experimental condition. Another thing that people often don't keep in mind when they're doing these membrane potential measurements is that the accumulation of the probe in mitochondria does depend on the inner mitochondrial membrane potential, but it's also going to be affected by other characteristics of mitochondria. For example, the amount of mitochondria per cell, mitochondrial mass, the composition of mitochondrial membranes, and the morphology of mitochondria are going to affect the accumulation of these probes. And that means the changes in the amount of probe per cell are not always because of changes in mitochondrial inner membrane potentials. And this is something that I really want to stress here. So this is a cartoon in which we look at situations of cells in which the mitochondrial inner membrane potential is exactly the same, but we have changes of accumulation of the probe in the cell for different reasons. So this first situation here is a cell that was loaded with one of these membrane potential sensitive probes, and is a cell that has a plasma membrane potential of 60 millivolts, and therefore the probe is going to accumulate 10 times in the cytosol versus outside the cell. And we added 1 nanomolar of the probe outside the cell. So within the cell, we have 10 nanomolar of this probe. Now, if the mitochondrial inner membrane potential is about 180 millivolts, we can calculate this through the Nernst equation. The probe is going to accumulate by 3 orders of magnitude within these cells. So it's going to be 10,000 nanomolar or 10 micromolar within mitochondria. So you can see here how much these probes really accumulate in mitochondria. That's why they can have toxicity. Now, this is situation A. Let's look at situation B here, in which the mitochondrial inner membrane potential is still exactly 180 millivolts. So there was no change in mitochondrial inner membrane potential. But what we have is a decrease in the plasma membrane potential to 40 millivolts. If you calculate how much probe is going to be in the cell under these conditions, you have around 4.6 nanomolar probe within the cytosol under these conditions because of this different plasma membrane potential. Because there is less probe within the cytosol, there's going to be less probe within mitochondria. So you're going to have a very steep decrease in the amount of probe per cell because there was a decrease in the plasma membrane potential, not because there was a change in the mitochondrial inner membrane potential, which continues to be exactly the same thing. Another situation that can change the amount of probe per cell without changing the mitochondrial inner membrane potential is a change in mitochondrial volume. So here you have a cell that has exactly the same plasma membrane potential and exactly the same mitochondrial inner membrane potential, and therefore you have exactly the same accumulation in concentration of the probe and mitochondria as the situation in cell A. But what happens here is that you have less mitochondrial mass per cell, so there is less probe accumulated in mitochondria in total, even though the concentration within mitochondria is the same. And once again, you're going to have a decrease in the amount of probe per cell in a situation in which the mitochondrial inner membrane potential is exactly the same. So that can generate a lot of confusion in the literature because people often equate the amount of probe that they're detecting with a change in the mitochondrial inner membrane potential, and that's not always true. Now, adding to this complication in which the amount of probe per cell doesn't always equate the mitochondrial inner membrane potential, there is another added problem, which is that the detection of the amount of probe per cell can also change. So you're not always detecting a direct relationship between fluorescence of the probe and the amount of probe per cell. You can actually have changes in the detection of the probe per cell, in addition to this accumulation of the probe per cell that also changes. So let's talk a little bit about how these inner membrane potential probe fluorescences are detected. And here there are many different ways to detect the fluorescence of these probes in the literature. You can detect these probes with high-resolution microscopy at a subcellular level in which you're actually seeing the probe within mitochondria and around mitochondria, or even within the individual cristae of mitochondria. And this is actually a really interesting experiment to do because you can quantify the inner mitochondrial membrane potentials in mitochondria by looking at how much probe you have in the mitochondrial cristae or in the mitochondria and how much probe you have outside, and calculating through this relationship what the inner membrane potential is. More commonly, people will detect whole cell fluorescence with lower resolution microscopy, or even whole cell fluorescence through flow cytometry, looking at how much probe fluorescence they're detecting per cell. Also, we can see in the literature, perhaps a little less commonly, people will measure fluorescence of the whole cell suspension or of the whole cells at the bottom of the plate using plate readers. So here you're really looking at macro and not one cell at a time fluorescence. I really have to caution people using flow cytometry or plate readers to measure mitochondrial membrane potential probe fluorescence, because you really don't know where the probe is. I think you should do at least some experiments to see if the probe is locating to a mitochondrial location, and making sure that you're actually seeing the probe at the concentrations that you're using. There are a lot of artifacts that can happen if you just throw these probes on cells and don't look at their loading and then measure their fluorescence through flow cytometry. So let's talk about measuring fluorescence in intact cells when you're measuring at a mitochondrial resolution and then at a cell resolution. So if you're looking at individual mitochondria within cells and their membrane potentials, you actually want to see the fluorescence within these mitochondria. So you should use these membrane potential probes at very low concentrations that avoid quenching. What is quenching? Quenching happens when you have so much probe accumulated within the mitochondrial matrix that the fluorescence of each individual molecule is quenched by the presence of so many of these molecules. So you actually have less fluorescence that you would have if all this probe were just suspended in water. So the individual molecules of this probe have their fluorescence quenched. And you actually can't see the fluorescence when you have overt fluorescence quenching, when you have a lot of these probes accumulating in mitochondria. You do not want fluorescence quenching when you're looking at mitochondria with a mitochondrial resolution, with high resolution microscopy, because you want to see these mitochondria fluoresce. So you have to use very low probe concentrations. And the amount of probe you're going to need is going to vary depending on your cell type, depending on the amount of mitochondria you have per cell. So you really have to titrate this and try a lot of conditions to make sure that you're avoiding this quenching probe concentrations. Second, you have to be aware when you're measuring mitochondrial inner membrane potentials at a mitochondrial level, at very high resolution microscopy, that mitochondria move around within the cell. So there's going to be motility, and if you stop seeing fluorescence of this probe, it can be because you lost your inner membrane potential, but it can also be because your mitochondria moved and you don't have mitochondria there. So one thing that can help a lot is to have a second mitochondrially located probe that is not membrane potential sensitive, so you know where your mitochondria are and you're aware of this mitochondrial motility. Finally, a thing that is done a lot in the literature that really bothers me is that people will express membrane potentials as a percent membrane potential decrease when they see the same percent decrease in fluorescence. And as this is often the case if you're looking at other parameters, for example, if you're quantifying a protein within the cell and you see a percent decrease in the detection of an antibody that binds to this protein, this can be related to a percent decrease in that protein. Now, when you're talking about inner mitochondrial membrane potentials, this is not true, because these probes are going to accumulate in mitochondria on a logarithmic basis related to the mitochondrial inner membrane potential. You can calculate this through the Nernst equation. So a decrease of 5% in fluorescence is not a decrease in 5% in membrane potentials. And when you're looking at mitochondrial resolution, you can actually calculate in millivolts how much the membrane potential has changed. So you do not need to use these percent expressions. You can actually do these calculations. Now, one thing that's really important if you're quantifying membrane potentials in millivolts, and I really want to stimulate people to do these quantifications, they're really important. When you're doing these quantifications in millivolts at a mitochondrial resolution of membrane potentials, you should not subtract backgrounds. Because if you subtract backgrounds, you're also going to have a quantitatively wrong measurement of the mitochondrial inner membrane potential. So let me exemplify this again with a cartoon. Here we have an image of a cell that's accumulating a membrane potential probe. So you have a change in the amount of probe in the cell and outside the cell. And if you do a cut of this image, you can see that there is more probe within the mitochondria within this cell versus the cytosol. So here we're cutting across not the whole cell, but this is an image of the mitochondria within the cell. The first thing that you have to be really careful about is that the resolution of your microscope is not being saturated by the fluorescence of the mitochondria within the cell. So we can see here that the probe concentration is increasing in the part of the cell that has the mitochondria on. And we want to make sure that the fluorescence is increasing proportionally and not much less fluorescence increase, because this part of the probe concentration has saturated within your microscopy measurement conditions. So make sure that you're not having saturated images in this sense. Another thing you want to make sure is that you can quantify the probe inside mitochondria versus outside mitochondria, because then looking at the relationship between these two concentrations, you can calculate the correct inner mitochondrial membrane potential. Now, if you get this image and subtract a background, you're going to be subtracting color from both inside mitochondria and outside mitochondria. And if you subtract color from inside and out mitochondria, you're changing this relationship, this ratio between the concentration of the probe inside and outside mitochondria. And that's going to give you an incorrect measurement of the inner mitochondrial membrane potential. So subtracting backgrounds is quite common in microscopy and fluorescence microscopy. But when you're calibrating inner mitochondrial membrane potentials, you do have to calculate this probe inside and outside mitochondria without doing these background subtractions. Otherwise, you're going to get the wrong numerical quantification of the inner mitochondrial membrane potential. Now, that's membrane potential detections at a level of mitochondria within the cell, looking at high resolution microscopy and quantifying the amount of probe within mitochondria and on the outside of mitochondria. What is more common is to do membrane potential measurements at a cellular level, looking at the whole cell fluorescence. And here really I have to caution you again that if you're doing this, and particularly if you're doing this with cytometry techniques in which you're not looking at the loading of the probe within the cell, it's really problematic and you have a high chance of having artifacts in your measurements. Specifically, for example, you can have artifacts in which the accumulation of the probe is really not in mitochondria, or if you don't know if you're at a concentration of probe in which you have quenching of fluorescence in mitochondria or not quenching of fluorescence in mitochondria. Another thing that I have to caution you about when you're doing measurements at a cellular level is the role of the plasma membrane potential versus the inner mitochondrial membrane potential. And I showed you how the plasma membrane potential can change accumulation of the probe within the cell, and therefore accumulation of the probe in mitochondria independently of changes in the mitochondrial inner membrane potential. Another thing that's absolutely necessary to know when you're measuring things at a cellular level is if you have this probe at a concentration in which fluorescence is quenching in mitochondria, or if you have this probe at a concentration in which fluorescence is not quenching in mitochondria. This is important because if your probe is in quenching mode, when you have a high membrane potential, you have less fluorescence in the cell because all this probe is accumulated and not fluorescing within mitochondria. That means if the mitochondrial inner membrane potential decreases, this probe is going to be liberated into the cytosol where it fluoresces again, and you're going to have an increase in fluorescence with a decrease in mitochondrial inner membrane potentials. On the other hand, if you have very low concentrations of probe and it is not quenching within mitochondria, If you have a decrease in mitochondrial inner membrane potentials, you're going to have a decrease in probe concentration in the whole cell, and therefore you're going to have less fluorescence in the cell. This can be quite confusing because often authors don't know if the concentration they're using is leading to quenching or not quenching within mitochondria. Now, it can sound really complicated that there's quenching of these probes within mitochondria, but this actually can be really useful for measurements in whole cells. To use the probe in quenching mode can actually help differentiate between changes in the plasma membrane potential and changes in the mitochondrial inner membrane potential. So here we have an example in which we have cells loaded with very low concentrations of probe. So they're in non-quenching mode, and this probe is fluorescing within the mitochondrial matrix. In this case, if you remove the plasma membrane potential, for example, using potassium outside the cell, you're going to have a decrease in the whole cell fluorescence because you're going to have less probe within the cell because of less plasma membrane potential. On the other hand, if you decrease the mitochondrial inner membrane potential, adding antimycin A, which is a respiratory chain inhibitor, and oligomycin, which is an ATP synthase inhibitor, you're also going to have a decrease in the whole cell fluorescence because now mitochondria are accumulating less probe, and that is fluorescing less in the whole cell. So you can't differentiate between a decrease in mitochondrial inner membrane potential or a decrease in plasma membrane potential. On the other hand, if you have the cell loaded in quenching mode, in which there's so much probe within mitochondria that its fluorescence is quenched, if you add potassium to remove the plasma membrane potential, you're going to have a decrease in fluorescence because there's going to be less probe in the cell, less probe in the cytosol where it fluoresces. If you add inhibitors of the mitochondrial inner membrane potential, antimycin and oligomycin, the probe that is quenched within mitochondria is now going to leave mitochondria, go into the cytosol, and lead to a temporary increase in fluorescence, which is then going to decrease over time as this probe now re-equilibrates across the plasma membrane potential, which still has its membrane potential. So here in quenching mode, you can actually differentiate between something that decreases the mitochondrial inner membrane potential versus something that decreases the plasma membrane potential because they're different responses. So quenching mode is actually really useful if you know that you're working in quenching mode, versus non-quenching mode that can also be useful if you know that you're working with it, but in which it can be difficult to differentiate between changes in plasma membrane and inner mitochondrial membrane potentials. If you're a bit confused about this and you want to read a little bit more about quenching versus non-quenching modes, I really have to recommend in general in mitochondrial bioenergetics the book by David Nichols and Stuart Ferguson on mitochondrial bioenergetics. They talk a lot about quenching and non-quenching modes. And David Nichols actually has a number of papers in the literature in which he uses these different probes in cells in real life to measure, quantitate mitochondrial inner membrane potentials under different conditions. And he talks about this and tries to help you through this. But what I really hope that you take home from this class specifically is that mitochondrial inner membrane potentials, without all this calibration, without all this care to know exactly what mode you're measuring in and how you're measuring this mitochondrial inner membrane potential, it really is not a sensitive or good way to assess oxidative phosphorylation in cells, because you have all these caveats and you have all these other effects that are going to change the accumulation and the fluorescence of these inner mitochondrial membrane probes. So if your interest is to measure oxidative phosphorylation, I cannot stimulate you enough to, instead of buying a kit and measuring fluorescence of these probes, to try to seek a way to measure oxygen consumption in your cells. Because this is a measure of flux, it's going to be a lot more sensitive, because this is a direct and quantitative measurement and is a lot less artifact prone than mitochondrial inner membrane potential measurements. There are a number of equipments around that can measure oxygen consumption in different conditions in different cells. And they're also available quite easily in most large research centers as analytical facilities. And in fact, if you are in my region, in the region of São Paulo, Brazil, we offer these different instrumentations to measure oxygen consumption as a core facility or as an analytical center in which you can come do these measurements, pay charges that just are sufficient to upkeep these instruments working in our facilities, and actually do these measurements in a much better manner. And feel free to contact me if you're interested in doing these oxygen consumption measurements. And I really have to end by thanking Fernando Abdulkader, who is my colleague and friend, because this class is very strongly based on a paper that we published earlier this year in the Biophysical Journal, which talks about how and when to measure mitochondrial intermembrane potentials. If you have any doubt about this and want to read the full paper and don't have access to the full paper, you can also contact me. I'd be glad to send you a full PDF of this paper, and also to work with any doubts you have about mitochondrial intermembrane potentials. So thank you.